This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. It's time for This Week in Microbiology, episode number 56, recorded on May 20th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast all about unseen life on Earth. Today, we're coming to you from Denver, Colorado. We're at the 2013 meeting of the American Society for Microbiology, and we're broadcasting in front of a live audience. Hey, everybody. And we're showing the audience what it's like to record a live podcast. We do this every other week in our offices. We try and bring microbiology to everybody. So we've selected some of the interesting microbiology here at the conference, so we're going to chat with them and see what is it that they do. I have most of the TWiM crew here with me today. On my left here from the wonderful blog, Small Things Considered, Elio Schaechter.
Hey.
Nice to see you in person.
Nice to be here in vivo.
In vivo, that's exactly it. Thanks for joining us today. And on my right, from the Medical University of South Carolina, also known as Dr. Copper, Michael Schmidt.
Hello, Vincent. Good to see you, Elio.
Nice to have you here. Thanks for coming. I'm sorry it's a little tight in there. All right.
We fit?
We do fit. Now, we've picked 4 guests. This is a big panel here. It's going to be interesting to see how we do this. We have a really wide-angle shot here. These are 4 people who are talking at the meeting, and we picked them, and we thought we'd highlight what they're doing. So all the way on the right there, a professor at Tufts University School of Medicine, Andy Camilli.
Hi, everyone.
Welcome to the show. Can I call you Andy? Is that all right?
Yes, you may.
Thanks for joining us. To his left, a professor at the University of California, Berkeley, Suzanne Fleiszig.
Hi, everyone.
Welcome. And I pronounced it right. Is that right?
That's
Fleiszig.
right.
You gave me a good clue. Over on the other side, to Elio's left, a professor at the University of Washington, Ferric Fang.
Hi, everyone. It's
Welcome to
Thank you,
TWiM.
Vince.
And all the way on the left, professor at the University of Michigan, Michele Swanson.
Hello, Vincent.
Welcome to TWiM.
I'm delighted to be here.
And what I would like to do, before we start the actual show, I have a couple of things to tell folks. We do have a mic back there, so if you are brave, you can get up any time during this proceeding and ask a question, all right? Don't be shy. You can get recorded for posterity. So this is being broadcast to millions and millions of viewers, and it's being recorded and will be forever on the internet, or as long as the internet lasts.
I gotta say something, though.
You can't.
I can't.
Yes, go ahead, say something.
My old university, Tufts University, is extremely well represented here. In the audience is the president, the current president of the society, Jeff Miller, who was one of our best graduate students. We looked at him and we said, shows promise.
All right.
Andy is on the faculty, and Michele was a postdoc in the lab of Ralph Isberg, and I was on the faculty there.
Well, one of the things I'm going to do in a minute is find out where everybody has come from to see how many degrees of separation there are, which aren't very many usually. If you are out listening on Twitter, you can send in some questions. Use the hashtag ASM Live. And Jim back there will be monitoring that stream, and he'll periodically ask the questions as well. So you can interact with us even though you're far away. Anything else I needed to say? I think that's it for now. Now, I want to just go around to all of our guests and tell us how you got to where you are today. I want to start with where you were born, high school, college, PhD, and all of that. Let's start with you.
You got all night?
Yeah. No, it'll be fine.
Yeah, how many minutes do I get? So I grew up in Michigan, and I went to the University of Michigan for my undergrad. And I started as a computer science major, but I switched to something more interesting, microbiology. I went to grad school I started at WashU in St. Louis, Washington University. And after a year, my advisor upped and moved. So I had to transfer to University of Pennsylvania in Philadelphia. and that's where I got my PhD degree from. And then I moved up to Boston to Harvard Med School for a postdoc with John Michelanos, and then got a job right across town.
Did you know Elio at Tufts?
Elio was just leaving, just retiring as the chair of the department when I moved in. In fact, I got his lab and office
And my couch.
his couch and his letter opener. And a funny story is about 12 years later, he visited me. I'm in my office, and I have the letter opener, and he says, I used to have a letter opener just like that.
Yeah.
He also left me a tremendous amount of glassware that filled all the drawers in the laboratory.
It's nice, isn't it?
Terrific.
All right. Suzanne.
So I grew up in Australia, as you can probably hear, and in Melbourne. I went to the University of Melbourne, where I did my original profession. I'm actually an optometrist, so I'm an eye doctor, and I did some clinical practice, and then I became really interested in why contact lens wearers were getting eye infections, and so that's what took me into microbiology. So I did a PhD working on the problem of why contact lens wearers get infections, and so that drew me into Pseudomonas research, looking at bacteria and why they infect and why they normally don't infect, and went on to do a postdoc in infectious disease at Harvard with Jerry Peer, focusing on Pseudomonas. And a few years later, I got the job at UC Berkeley, School of Optometry, as a professor.
All right. Berkeley. Ferric, where are you from?
So I'm a native of Southern California. I was born at UCLA and product of LA City Schools.
Okay.
And then I went to college and med school at Harvard and decided initially that I wanted to become an internist. So I came down back to California to UCSD to do a medicine residency. But I found that medicine as a specialty had evolved to become really more about economics and how to most economically deliver medical care. And that wasn't really what my primary interest was. I was most interested in physiology, pathogenesis. And I found that most of the interesting cases that really grabbed my attention, that really required detective work, were the infections. And San Diego is really an interesting city from an infectious disease point of view because there are a lot of immigrants and a lot of recent immigrants and a lot of infections that you don't normally get to encounter studying in the U.S. So I gravitated to infectious disease, and I was happily on my way to becoming a practitioner where I imagined myself in a white coat with an admiring throng of medical students following me around, and unexpectedly killing off the research part of my fellowship, I found that I really enjoyed life at the bench. And I did 2 postdocs to kind of make up for not having done graduate work. And this allowed me to have at least enough working knowledge to be dangerous and to go off and start my own lab when I joined the faculty at University of Colorado here in Denver. So I was here for 9 years, very happily, with great people. And then moved 12 years ago to University of Washington. My other sideline, in addition to doing then molecular bacteriology research, is diagnostic microbiology. I oversee a clinical microlab at a busy county hospital. That's about half of my time. And then I've had the privilege of being an editor at Infection and Immunity, and then for the last 6 years, editor-in-chief. And so this has turned out to be a really interesting part of my career because I can take a look not only as a scientist, but as someone who's very interested in science in general, in the process, in the enterprise. And with my good friend Arturo Casadoval, who's the editor-in-chief of MBio, I've had a really interesting time thinking over things with him about how science is right now, how it's practiced, if it's really as good as it could be, if it's really serving its mission as well as it could. And this has been something that I've been spending more and more time thinking about.
So we've actually talked about some of that work on my other podcast, TWiM, and today I hope to talk a little bit more about some of your more recent work in that. So Michele, tell us, I know part of your history, but tell us anyway.
So I grew up in a large family in the Midwest. I went to high school in Ohio, and I was a 3-sport athlete. my field hockey team went to a camp in Michigan, and I met the field hockey coach at Yale there. And a year later, I contacted him and asked for advice about where I could go for college, where I could get strong academics and a good program. And he said, apply to Yale. So I did, and that started me on a life on the East Coast. So I was a biology major at Yale, and I played field hockey and softball. And then I never stepped foot in a lab as an undergrad. But for my first job out of college, I got a job as a technician at Rockefeller University. And this was in the department of the late Zancone with Sam Silverstein. And they were doing what we now call cellular microbiology. So I got my first taste of the interaction of microbes and white blood cells as a technician. In fact, Marcus Horowitz was doing some of the pioneering work on Legionella at that time as a fellow, and that's the bug I work on now. I then started graduate school at Columbia, and I took a very influential virology course from a young, brand new assistant professor, Dr. Vincent Racaniello. Learned some molecular biology from him. I then transferred to Harvard because my husband got a faculty position there, so I started with Marion Carlson at Columbia, and then did most of my work with Fred Winston at Harvard. Toward the end of my PhD, I wanted to do something that felt a little bit more relevant to human health, and I was fortunate to get a postdoc with Ralph Isberg. And there I could apply my experience with classical genetics and molecular genetics and cell biology to study the pathogen Legionella pneumophila, which grows in macrophages. So I got a faculty position in 1996, moved with my two kids to Ann Arbor, and I've been there, I think, 17 years.
What was the third sport?
In high school, I played field hockey, basketball, and softball.
Okay. Do you do any of that anymore?
I play golf now, and I'd love to talk about it.
As all golfers do.
Right?
Warning.
So I love to hear where people come from, because you see all the interconnecting threads here. And that's the way science is. It really is a small field despite having so many people in it.
Right.
So sorry, did someone say something? Did you? I'd like to start with you, Ferric, and talk about two of your recent papers, one about misconduct and retracted publications. And this is not something that all scientists like to talk about, but I think it's good to get it out there and talk about the problem. So I'm curious why you started to do this study in the first place.
So I think was like most scientists several years ago in thinking that misconduct was quite an unusual event and probably an aberration due to the individual psyche of the person who got themselves into science, but for whatever reason was motivated to do things in an unethical way. And I thought it was so rare as to not be worth thinking very much about. And I also thought that there's an old saying that everyone knows that science is self-correcting. And so I had faith that the process of science would root out anything that was dishonest or incorrect, sloppy, erroneous. And in the end, the truth will out. And I don't think that's completely false. But I think it's a little bit too simplistic. And what changed my view was really my experience as an editor-in-chief, where one evening I was sitting in my office and an email popped into my inbox. And I'm really compulsive about checking my email. So even though I was about to go home, I took a look, and it was from a dean of a medical school in Japan. And the letter said, "I'm very sorry to trouble you with this, but one of our faculty has been accused of research misconduct. And the investigation has fallen to me and a committee that I've assembled. And we've reviewed all of the papers that this individual has published during his career over 10 years at our school. And several of them involve your journal. And because they involve your journal, and because our finding is that the data have been manipulated and misrepresented, we felt obligated to notify you." And so there were several papers from I&I, as well as from other papers at other ASM journals and many other journals. And this was the beginning of my baptism into pathological science. And in the end, we ended up retracting all the papers published by this individual, and we found that he had engaged in this repeated behavior of taking things like the picture of the same gel and reproducing it in different articles, claiming that it was one thing on one occasion, another thing on another, cutting bands and moving them around, and using control bands that were from one experiment and representing them as controls for a gel from another. And it ended up 31 papers so far from this lab have been retracted. And it occurred to me that...
Is that a record?
No, actually not close, unfortunately. But it was quite startling to see that these papers had been in the literature all this time, that people had been reading them, downloading them, using them, photocopying them, citing them hundreds of times, and no one had corrected them. One of the things about correction is it assumes someone's going to try to do exactly what you did. But a lot of times that doesn't happen because we're trying to do something new in the lab. So when we find someone's already done something, we try to find something else to do. Another problem is that he was very clever in getting experimental results that were very likely to be true, but he wasn't really showing them. He was falsifying data in order to get the result he expected. But the results weren't very surprising. And so nobody stopped to say, well, that's probably not right. If you say something that is startling to everyone, like there's arsenic in DNA, then everybody's going to be very interested in that, and they're going to be running out to check, and that you're going to have lots of different people chiming in right away. And in this case, science works, I think, in the classical way. But there's other stuff buried in there, I think, in the literature that are not really rigorously done or not even honestly done, and it won't really be found out unless you get lucky. Many cases, there's a whistleblower, but in this case, what happened is a very astute reviewer, who I actually have had a conversation with, but I don't know if she wants to have her name revealed, so I won't. But she contacted me after reading about some of these things and said, I'm the one who found this. She said, I'm in this guy's field, and I was reading a paper that I was asked to review, and I had a deja vu experience. And I thought, this looks familiar. I don't understand this. There's a new paper being submitted. And she started pulling papers off the shelf, reprints. She had a student come in. She said the office was covered with this guy's papers, opened up, and they started seeing all of these patterns. And they notified the editor of that journal, which was Blood, who then investigated, notified the institution, and then the cascade of events eventually led to me. So this changed my view, and in thinking about what these articles meant, I started looking more carefully at the process when there's a suspicion that an article has an error or is false, how this all works, how journals handle it. And I was kind of surprised to see that it's really quite an ad hoc process. There are no hard and fast rules. Journals can do whatever they want. Journals don't investigate misconduct. They may have a suspicion that there's a problem with data, and as a result, they may attempt to find out whether there's a concern that the data are legitimate. But in terms of actually going to get raw data and interviewing people, trying to get at the bottom of something, that really has to be done by the institution. So the journal may raise an issue of a possible problem to an institution, but then the institution has to investigate it. This can take a very long time. In the meantime, the journal has to decide what to do. Will we retract the paper? Will we express concern about it and let people wait and see what happens? If the paper is retracted, how much information should we provide? Do we provide a detailed blow-by-blow? This figure is suspicious because it looks exactly the same as a figure from another paper? Or do we simply say the paper is withdrawn? Or do we say it's withdrawn due to suspected data falsification? It turns out that the practices are widely variable. And everybody was talking about what retractions mean. There's a blog, Retraction Watch, that's become very popular, run by Ivan Aransky and Adam Marcus. And every paper that's retracted, just about, they write a post on it. And then people come in and comment on it. And this creates lots of news interest. And so it's become a topic of a lot of interest. And it occurred to Arturo and me that there weren't a lot of data. And so we tried to go through selected data sets, and we realized no one is really going to take this seriously unless we look at everything. Mm-hmm. So for 1 week in May, we downloaded every retraction from PubMed, which is over 20 million articles.
How many retractions?
And we found over 2,000 retracted articles.
Okay.
And then we started looking over every retracted article and the retraction notice. And we found that in a large number of cases, over 100 cases, the retraction notice sounded like it was a mistake. But when you dug down and you looked at the other information, sometimes the New York Times or the Washington Post, sometimes the report from the Office of Research Integrity, which investigates anything supported by the Public Health Service, we would find that there was actual misconduct. And so we started going through, we made this huge Excel spreadsheet, and we started reclassifying things. And when we did, we found that existing surveys of the literature, which said most retractions are due to error, were actually wrong, that most retractions were due to data falsification or fabrication, or due to plagiarism, duplicate publication, and so on. So misconduct of some sort. And that's what led to the article last fall in PNAS.
So is the rate of retraction increased in the past years?
So the rate of retraction has clearly increased. No one questions that. What is debated is misconduct more common, or are people catching it more? Because there are more people looking at it. You tend to find more retractions in highly read journals with high impact factor. More eyes are on them. A lot of retractions for flawed data involve digital data. It's so easy now to manipulate data with Photoshop. So there are lots of do's and don'ts about preparing data for publication. And so in some ways it's easier to do, in other ways it's easier to detect. There are plagiarism detectors now, and people run papers through. They'll also use programs like to find duplicate publication. So I think that both are going on. But one of the points I would make, before I'd like to let other people comment, is that if misconduct, as we look at the retractions for misconduct, they've gone up tenfold. So if you're going to argue that that's all detection, then you have to say going back 20 years ago, when people like Elio were running a laboratory,
At least.
that 90% of that stuff that's due to misconduct we haven't caught yet.
Don't look at me.
I think that's very unlikely, and I don't think there's any evidence to support that. So I do think the incidence of misconduct in the literature has gone up. The other thing is how much misconduct is actually happening. We can look at retractions as a little piece of misconduct, and the way I choose to look at it is as a symptom of science that's gone wrong. That a retracted paper, whether it's for error or for misconduct, is a tragedy. A lot of money goes into that, about $250,000 as far as we know per paper that gets retracted. So it's a huge amount of resources. All the people who go into all their efforts, you know how hard it is to get a paper. It's like having a baby. And when it finally comes out, everybody is so happy. And then to have that all come apart with the retraction, it's humiliating for everyone. And it's disruptive for the scientific process. People have cited that paper. And so even if it's retracted, it's embedded in the literature. That information is already disseminated. So I look at it as a symptom of a process that didn't work in that case, and try to understand why it didn't work, and therefore gain some insight into what goes on in science. The other thing I would ask my colleagues here in the audience is, how common do you really think misconduct is? Because if it's only retractions, that's 1 out of 10,000 papers. That's very rare. But if you survey scientists anonymously, you get numbers more like 2 to 3% have seen serious misconduct. And then there's all this squishy stuff, like taking out a data point or not reporting the times that the experiment didn't work, and the kinds of fudging about things, the use of the wrong analytical test. I heard today at the I&I editorial board meeting from one of my editors. She was on a committee meeting for a graduate student, and she looked at the statistical test applied to the data, and she said, "I've never heard of this test. How did you choose it?" And the student said, "I got onto the statistical program, and I checked the p-value by all these different tests, and this was the only one that came out less than 0.05. So there's lots of other practices which might not be flagrantly dishonest, but are clearly suboptimal science. And when you look at it from that end, you start saying, this is not actually a fringe problem. It's a problem with a significant part of science that it's worth scientists thinking about.
Let me ask you, what do you think about the self-correcting quality of science? We know that when somebody makes a claim which is very important sounding, that claim is going to be tested. That claim is going to look at the arsenic story. I mean, it didn't take very much time to figure that out. If somebody makes a claim which is so remote and so peculiar and so special, nobody cares. So damage has not been done if you report that you found a strange bacterium in the left nostril of a rhinoceros or something. Who cares? But if you make a claim which is pertinent to today's science, self-correcting does play a role.
So let me, I don't completely disagree with what you say, but let me give you a counter anecdote. Few would argue that some of the most important research that we really rely upon to be robust are research that we would use to influence clinical healthcare. And yet, we had the scandal at Duke where there were these papers that were generated that were being used to guide cancer chemotherapy. Mm-hmm. Patients were actually treated, and the therapy didn't work. And now companies were created, and the whole story has fallen apart. Or we have the paper from Amgen in Nature where 53 studies that were published in high-profile journals as being the basis for new cancer drug discovery programs. Only 7 of them could actually be replicated by the Amgen scientists. So if we have a rate of over 80% of data that cannot be sufficiently duplicated to actually be used in the way that it's intended, then I think we have a really serious problem. So the truth is probably somewhere in between those two extremes. I think a lot of what we rely on our own fields does get replicated, because if I want to build on something somebody's done, I don't just take their word for it. I build off the end of what they did, and I try to make sure I can see what they saw. Right. And sometimes that doesn't happen. We have a big controversy going on now with, do antibiotics kill by reactive oxygen species? This is hugely important for drug discovery, and it's a very interesting problem from a scientific point of view. And we have excellent scientists on both sides who are heatedly disagreeing. So I think out of that, it's going to be sorted out in the classical scientific way, and we'll come out with a better understanding than before. So a lot of times it can work, but I also think we can't be complacent that everything that's in there, because nobody's retracted it or challenged it, we can assume is true.
Do you think that this is an appropriate use for supplemental material, where they really write a proper method section, where people can replicate their work, and more importantly, answer questions as to how many times they've conducted the experiment, how many times the experiment worked, effectively airing the dirty laundry inside the supplemental material? Because part of the problem with high-impact journals is the 3,000-word limit. And methods, let's face it, are the first thing to go. And when you begin to talk about the finer points of selecting a statistical methodology, you have to expend a substantial number of words to argue why you selected a particular statistical method. And it's sort of like inside baseball. Unless you're a statistician, you don't really appreciate the finer points. In that graduate student's statement, I picked the method because it was the best p-value. The statisticians are pulling their hair out, screaming, but people don't know what they don't know, and that's why you have a statistical consultant on many of these clinical trials, and they have taken great pains in selecting the proper method. And maybe that's a really good use for the supplemental section, so people can actually then validate rather than putting in badly edited results and badly crafted figures that typically are now going into the supplemental sections.
So I think you make some very good points, and I note that the ASM journals have a pretty flexible supplemental data policy. It started out as a place to put huge data sets or movies, things that couldn't go in the article. But it's turned out to be an important place to put detailed methods, full lists of strains, genotypes, things that people who are going to try to use those data in their own work will need to have. And I think that's a very appropriate use of supplemental data. Another movement, which hasn't caught a lot of traction yet, but there's talk about it, is having raw data in a repository somewhere. If you look at a gel and it's all fixed up, you might like to see the photograph of the original gel it came from. You might like to recalculate the statistics yourself if you had the Excel file with all the data in it instead of relying on the graph that's in the article. Certainly, editors of journals have the prerogative and use it to ask for raw data, but the reader doesn't usually have that prerogative. And I think there may be a time where that becomes much more standard, but at the moment it's mostly at the stage of conversation.
Do you have a sense of whether the rise in the number of retractions is continuing, or in light of your papers, has it abated at all?
It seems to still be at a very high level, but I'm not sure that it's accelerating. Mm-hmm. And I do think it's possible that it's starting to plateau. We've been trying to engage in some statistical analyses about the time to retraction, and my colleague Grant Steen, who was a co-author on the paper with Arturo Casadevall in PNAS, is very interested in using measures to create a mathematical model of the rate of retraction. Mm-hmm. And he's got some preliminary data he's been working on that suggested that maybe the rate is decelerating.
Okay.
And
we have a question.
Yep. Yes, question.
Hi, I'm Devin. I'm from the University of Maryland, Baltimore's dental school, and I just wanted to ask, with the bigger pressure nowadays for scientists and clinicians to publish or perish, and also with funding being cut, the sequestration, do you think that possibly could affect retractions and also falsifying of data, trying to get more publications out because, hey, I could get more funds possibly or something like that?
Well, I think that's very insightful, Devin, but it's because you and I are in complete agreement. So take that with a grain of salt. I mean, we're clearly all working under the same pressures, and the vast majority of scientists who I know are incredibly careful and honest. But at the same time, so I don't think it's one factor, but I do think that you have a situation where people are under enormous pressure to get things out, for certain deadlines. You have grant deadlines, and you want to get the paper published before the grant is submitted so that it will be a sign of productivity. There's a lot of pressure for people to publish in certain venues that have perceived high prestige. And so when you get into that situation, if you have somebody who's vulnerable, who might feel that they have so much at stake, that the psychologists and the behavioral economists in business schools have looked at this very carefully. What leads people to do something dishonest? And it's usually not an incentive of getting something. It's an incentive of losing something. And this strikes me, the psychology of loss, as something that I see all the time in science, where people are afraid of losing a grant, losing a job opportunity, and that kind of pressure, I'm not going to make tenure, I'm not going to have my grant, I won't be able to keep my lab together, is the worst kind of pressure that you can put on somebody in terms of getting them to behave ethically. And although the individual who commits a dishonest act is clearly responsible for that act, and I'm not letting anybody off the hook, I feel that as somebody who really wants a science we can believe in, we need to take a hard look at our system of pressures and rewards and how we're incentivizing people. Because if I tell somebody I want them to go out and simply find the truth, I have to create true incentives that I'm not going to penalize them if the truth is not what I wanted, if the truth is not a positive finding that's going to be published in one of those one-syllable journals. I think that right now, we PIs tend to speak out of both sides of our mouths. We want people to be honest, and we want them to work 24 hours a day, 7 days a week, and we want them to have balanced lives, and we want them to have a raft of high-impact papers. And so I think that we're all responsible for the research culture that we create at home. And one of the other papers I thought that Vincent might ask me about was this interesting paper we published in MBio a few months ago, where we looked at the people who were actually found to have committed misconduct, and we assumed we were going to find mostly trainees who were feeling this pressure, who had to get a paper out to get their PhD or to go on to get a job. And we were surprised to find that there were as many faculty as there were students and postdocs committing misconduct. These are the faculty who are supposed to be setting the example, creating the culture. And in many of the cases when we drilled down, we found that the students and postdocs, while they had committed misconduct, there was evidence they were working in a culture where they were under a lot of pressure. And so I think that the senior scientists need to take a hard look at the environment they're creating. We want to incentivize people to be successful, and we can't deny that we work in a world where we're being measured by our productivity. And on the other hand, we're all in science to really advance knowledge and to serve society. That's really what it's about, those two things. Not
It's the public trust.
Yeah.
It's the public trust.
And that's another interesting point, Michael, because some people have said, are you undermining public trust in us scientists by even bringing this up? But I would say, without us bringing it up,
We have to be self-policing, otherwise
aware.
the public will lose all faith, and it's very challenging to communicate very complex topics to the general public and for them to really appreciate the value that basic science brings. Everyone sort of gets applied areas, making a better something. But it's the basic data that are so important from which discoveries to the next best thing come. And if they don't have any confidence in our ability to conduct basic science, we won't see any funds. And if we don't police ourselves, then it's going to be, at our own peril.
Well, the public will find out about the misconduct anyway, whether we talk about it, right? And so the long-term effect is to undermine. We already have a problem with the public's perception of science and scientists, so this only makes it worse. So we have to address it. And, taking care of the reward system and the grant system is one way. But do you think early training in this whole issue helps at all? Now people who get money from NIH need to take a course in
You mean it's like hand hygiene for science misconduct?
Exactly. So
to teach us all to wash our hands.
We know hand hygiene is effective. Is that training effective? Do we have any way?
It hasn't been looked at rigorously. And one of the things that Arturo and I would like to see is more rigorous scientific self-analysis of the scientific process. And that's a question that could certainly be addressed more rigorously. But where it has been looked at all, for instance, Frank Macrina has been involved in medical ethics for a while, and he wrote a paper where they were unable to show a positive impact of training on whether or not the subsequent trainees were aware of and involved in proper research conduct. But that's not to say that it played no role at all. I think we would be naive to think that we could force students and postdocs to sit through 4 hours or 6 hours of lecture a year and that takes care of the problem. It's one little bit. What I find in my own lab is there are some simple things that we've started trying to do that I think make people more aware in a day-to-day way of how they do things. For one thing, I show them examples of inappropriate data manipulation and analysis from papers. And they see that this is happening and this is wrong. And a lot of people learn by example. If we have things in the literature that are wrong, that are analyzed by the wrong statistical test, that's a problem. But when 100 people copy that wrong way of doing it because they go to the literature to take that bad example, it's really amplified. So I try to use that, and we talk about ethics on a regular, ongoing, day-to-day basis. I always make sure that I look at the raw data with them when I look at things, not just the polished figure. And I ask them about how they statistically analyzed this and how they knew this was the best test. And sometimes it's not the best test. And we look at things together as a group, so the whole lab can interact. And I think it creates an environment where they understand that doing the right thing is really important and important to me. And it's more important to me that we do the right thing even if we get scooped, even if it turns out the hypothesis is wrong. We don't get that high-impact paper. I care deeply about their careers and moving them ahead, but I also care about training people who do science the right way that we're all going to be proud of. And I think, each of us has to do our part, and that in that context, having a research integrity series is a useful adjunct. But to think that that's taking care of the whole thing, I think would be naive of us.
Well, obviously, this is something we could talk about a long time, but we do have to move on. There are no more questions. So I want to thank you for seeding that little bit of interest. And everyone take that with you. And it's really up to us, in a way, to perpetuate that attitude. Let's move across all the way to the right, to Andy there. Get you a little engaged. And what I wanted to have you tell us about is this wonderful story from your lab recently came out about the finding of a CRISPR in a bacteriophage. And maybe you could start by telling us all what a CRISPR is to begin with, because we haven't really talked about it on TWiM, right?
All right.
We've been
waiting for you.
crisp.
Okay. Well, I only became a CRISPR expert about 2 months ago when we discovered, maybe 3 months ago. So my lab studies cholera, and we also study another pathogen called pneumococcus. But we study cholera, and this is a facultative pathogen, which means it lives in our intestine, but also it can live out in aquatic environments. And out in aquatic environments, the bacteria are exposed to bacterial-specific viruses, which are called phages. And it's long been known that there's this co-evolutionary battle going on between bacteria and their phages. It's been very well studied. For example, the discovery of restriction enzymes. That's a bacterial defense against phages. So it was actually discovered, the discoverer of phages, Felix de Herel, actually was the one that discovered that cholera patients, when they're shedding the bacteria in the watery diarrhea, outcoming with the bacteria are phages. He saw them as little clearing zones in a lawn of bacteria. So he discovered phages coming out of cholera patients long ago. And he surmised that these phages are actually killing the bacteria. So they're, in essence, our friends, because they're the enemy of our enemies. And I just read one of his papers from 1926, and I realized he's thinking the same things and seeing the same things that we are seeing right now. And I was very impressed by what he wrote. Anyway, so we've been studying the interaction of cholera and phages the last couple of years. And by chance, we discovered we were getting a hold of stool samples that had been frozen and archived from the past 10-year period at this one hospital in Dhaka where there's lots of cholera. And we did a survey. What are there phages present? What phages are there? And we were surprised when we discovered there was one phage that was omnipresent. It was in every patient's stool sample. Even though the cholera strain that would cause the outbreak that year might change, this phage was there. And so we immediately thought there's got to be something special about this phage to year in and year out tag along with the epidemic cholera strain. And so we started to study this interaction, and we discovered that just by sequencing these phages that they had a CRISPR-Cas system. What is a CRISPR-Cas? It's a horrible acronym. In essence, it is a way for bacteria, it's a phage defense system. Upon infection by a phage, the bacteria has a chance to capture little snippets of the phage's DNA and incorporate it into a locus in the bacterial genome. It's kind of like a library or a storage place for a memory of past infections. And what the CRISPR system does is it will express that little piece of DNA into an RNA molecule, and that RNA molecule will constantly be involved in surveillance for infection by that same phage. If that same phage infects that cell or its descendants, that little piece of RNA will recognize the phage DNA and essentially tack it, and there'll be double-strand breaks in the phage DNA, and the bacteria survives. So it's called an adaptive immune system of bacteria.
It has memory.
It has memory.
But not all bacteria have these systems. Right?
About half of bacteria have CRISPR-Cas systems. Mm-hmm. And interestingly, about 90% of archaea have it.
Why are you laughing like that?
The archaea were badly named. People think they're ancient, but they're actually much more modern than we give them credit for.
Yeah, they were born with calvos.
That's right. Yeah.
They didn't exist before.
So bacteria and archaea have these as defense systems against phages. Bacteria also have other immune systems to fight against phages, and what we discovered was a phage that has captured and commandeered a CRISPR-Cas system that bacteria normally use, and it is using it against the cholera bacteria. So the phage itself has an adaptive immune system, and it can keep step with what the bacteria are trying to use to interfere with the phage. So that was the discovery.
So how does that work? The phage DNA comes into the cell and it's transcribed by the host polymerase. So the host is
Yes.
taking part of it, makes the small RNAs, which then do what?
So it gets a little complicated. So cholera, some strains have an island of genes that's involved in resistance against this one phage. Upon phage infection, this island of genes excises from the chromosome as a circle and replicates, and it interferes with viral replication. And this has to happen very quickly. It's a race between the phage and the bacteria. And the phage uses this CRISPR-Cas system to attack and degrade this excised circle. And what we found is the phage CRISPR-Cas system is fully functional. It can, if you challenge the phage with a new excisable island that it doesn't have memory to, it'll capture a snippet of it and store it into its library, its memory, and it'll use it against the cholera bacteria. And so the phage, which already can out-replicate the bacteria and in many ways out-evolve it, now has an adaptive immune system. So we think that's part of the reason this phage has been omnipresent the last, 13 years as far as we know.
And if you take the CRISPR out of the phage, then it can no longer kill the bacterium, I presume.
Correct. If we delete the CRISPR-Cas system, the bacteria are now resistant to that phage. However, we've discovered that the same strain of phage has other mechanisms, and the bacteria have other defense systems. We're just now starting to explore this for this particular pair. But this has been studied very well for many decades in other systems.
So being a virus, the phage needs a host bacterium to replicate. So it's not to the phage's advantage to completely destroy all of the bacterial population. How is that homeostasis maintained since the principal bacterial defense seems to be inactivated by this phage?
That's a very good question. So one thing is cholera lives out in the aquatic environment, and if we put the phage together with the bacteria out in, say, pond water, the phage cannot kill the bacteria and replicate. That's because the bacteria themselves are highly challenged by the nutrient poor water that you place them in.
So they become lysogenized with the
No.
Vibrio?
No, this phage doesn't form a true lysogen. It doesn't do that. But what's more interesting, I think, is in the human intestine, when you ingest cholera and this phage, the phage is killing the bacteria while the bacteria are growing in our intestinal tract. And so we see some patients will come into the hospital with watery diarrhea, and they're very sick, and you try to culture cholera from the secretory diarrhea, and you can't detect any cholera there. That's because the phage, we think, are annihilating the bacteria. And we suspect that there's a lot of asymptomatic or very mild cholera infections. That's been known for a long time. And we suspect that phages may play a very important role in that, along with immunity, of course, host immunity. So phage therapy is kind of once again, a hot topic. And I would argue that phages have been helping humans for a long time, at least for cholera.
So why only this phage? If I were some other phage, I'd like to have CRISPR within me. Why don't all phages have CRISPR systems?
So some phages have a very small genome, and they just don't have the capacity to.
Yeah, but some are big.
Yep. So there have been reports from virome sequencing projects of other phages with either pieces of the CRISPR-Cas system or entire CRISPR-Cas systems. But nobody had shown that it's actually functional. So there may be other phages out there that have it, but by sequence databases, the vast majority do not have it. So that's a very good question. What we think is that this cholera excisable island of genes is like the perfect target for the phage CRISPR-Cas to be used against. It's probably not going to be that effective for other defense systems like restriction modification systems.
Any indication the CRISPR sequence may be bacteria derived originally way back?
So it's the phage CRISPR-Cas system is most closely related to the CRISPR-Cas system from Yersinia pestis. However, it's quite divergent from it. That's the closest homology. We suspect this phage is very specific for Vibrio cholera and only a particular type of Vibrio cholera. So we suspect that sometime in the past, we have some recent data that is probably at least prior to 1964, that it must have infected some environmental cholera strain and captured the CRISPR-Cas system. Interestingly, prior to the 1960s, the cholera strain that caused epidemic cholera and pandemic cholera had a CRISPR-Cas system. But in the 60s, that strain was replaced by the current 7th pandemic strains, which don't have a CRISPR-Cas. But that the old classical strain of cholera, its CRISPR-Cas is not the system this phage captured. It's a different system altogether. So we don't really know where the phage captured it from.
So you had mentioned phage therapy, but in this case, using this phage for therapy for cholera wouldn't work because of the CRISPR in the phage, right?
So the cholera that has its own CRISPR-Cas is thought to be extinct in nature.
Okay.
So I think this phage might be a useful component of the phage.
Do you worry that if we started to use phage therapy, that they would then start picking up CRISPR sequences from hosts that we were trying to kill with them?
Are you afraid of making the phages more
No, I'm not afraid of
successful?
anything. I'm not afraid of anything. I'm just wondering if you're thinking that that could be an issue.
Phages have plenty of opportunity to capture these things.
Yeah.
So I don't think humans developing phage therapies is going to represent any danger in that respect.
Well, that's the hallmark of the Elio Institute's program for phage therapy out of Georgia, is they are constantly isolating new phage and asking, will they work against the current group of pathogens that their cocktails are effectively designed for. And one of the reasons it hasn't made it to the U.S. is because of the whole way of developing ethical pharmaceuticals is they don't like things that are constantly changing. And the Elio is constantly retooling their phage cocktails to deal with this tour de force between the host and the virus. And they're going back and saying, okay, we need a new series of phage that can work against this crop of pathogens that we are seeing in our population. So it's a constant iterative step, and I think that's what we see with your cholera, because of the 7th pandemic strain is something new, and all of a sudden the phage says, well, we have to adapt or we're going to die.
To be devil's advocate, I'm not sure that I'm personally that enthusiastic about phage therapy with this phage for this disease, because I'm intrigued by your suggestion that maybe the self-limiting nature of cholera infection is really phage dependent. And it's been known for a long time that antibiotics don't play a huge role, even though we can eradicate cholera, because what we really need is the fluid and electrolyte replacement to keep the patient alive, and then they'll be okay. So I'm wondering if adding a phage therapy to clear the organism a little quicker really won't have a lot of clinical benefit beyond tetracycline.
So the current data suggests that a full-blown cholera infection, where you don't give antibiotics, you just let the patient's immune system respond, and you just give them rehydration therapy, that only generates about a 7 or 8 year immunity. So patients can get infected later on. But a big problem with cholera that's becoming more and more appreciated is that most of the transmission happens within households. So Stephen Calderwood and Firdousi Qadri are some colleagues of mine that have been looking at this closely. And when someone comes into the hospital in Dhaka with cholera, what they found is that about half the people in the household come down with cholera or seroconvert within 2 days. So transmission is happening in the household, and people get very sick and they die.
So phage therapy to interrupt transmission then might...
Right, so that's an idea.
Okay.
The alternative is you go give antibiotics to the household contacts, and that comes with its own problems where you're clearing the good bacteria. So
Yes, a question.
Amy from the University of Colorado, Denver. Along those lines about clearing out the good bacteria as well, can you comment on how these phage interact with what we consider helpful commensal bacteria?
So the question is, will phages against pathogens affect the commensal bacteria?
Yeah.
So that's a beautiful thing about phages is they're usually very specific, not always, but usually very specific for a particular species of bacteria and often a particular strain type. So for example, the phage we've been talking about with the CRISPR-Cas system only infects one tiny fraction of the species of Vibrio cholera and doesn't infect other bacteria. So in that way, it's perfectly designed to be very, very specific, which is not true of antibiotics.
So would you need some sort of molecular diagnostic to be able to tell what kind of cholera strain was involved before you used phage therapy?
So cholera is nice for this reason because it's really, there's only two serogroups that are caused cholera right now. O1 and O139 they're called. And so maybe if we started using phages against these, we'd have other serogroups move in to fill the void, as is happening with pneumococcus. But that remains to be seen.
Pandora's box.
So can you tell us the next step for this story, as far as you're interested in it?
So we think we pretty much have figured out how the phage uses this CRISPR-Cas system against the bacteria. What we're very curious about now is this inducible, excisable chromosomal island that mediates resistance to the phage. We're very interested in how that works mechanistically. And there's been some work done by Richard Novick's group, and gram-positives have elements like this as well that give resistance to phages.
So can resistance arise to the phage CRISPR sequence? Can it go back and forth?
We haven't seen that yet, but there have been some recent reports, and in fact some very nice talks here at this meeting on phages evolving resistance mechanisms against the bacterial CRISPR-Cas.
Different from the one you're talking about here.
So the bacteria has a CRISPR-Cas that uses for defense against phages. And people have now found phages that encode small open reading frames that make these small proteins that can bind to and interfere with the CRISPR-Cas functionality. So basically interfering with the bacterial's defense system.
The part of the CRISPR-Cas is a protein that is involved in cleavage, right?
Yep.
Of the target. And so these phage antagonist proteins can interfere with that.
Yep. So we don't know if Vibrio cholera has genes and proteins that can interfere with the phage's CRISPR-Cas. That we haven't seen yet.
Sounds like there's a lot left to be learned
I
here.
have a question.
Sure, go ahead.
No, there.
Oh, sorry, yes.
Hi. So back to the therapy. Does one consume a phage, or does one consume a bacteria containing a phage? How exactly do you envisage getting the phage?
The phage therapies
to the
that
right
have been
place?
phage therapies that have been used over the years, like in Russia, you consume a cocktail of pure phages, not the bacteria, and the phages will seek out and destroy the bacteria they're specific for. I should mention, by the way, that the FDA in the United States has approved several phage products that are used for example. A cocktail of phages that can be sprayed upon vegetables, and the phages are specific for Listeria monocytogenes. So we do have phages that are being approved as products in the United States, and of course people are, going to ingest these vegetables. So I think that hurdle has been overcome.
Yeah, and we've talked on a recent TWiM about propionibacterial phage specifically targeted for acne, because it's interesting in that the phage for acne will cut across all the propionibacterial strains, and the phage, one phage type is specific for a multitude of different strain variants of propionibacteria. So phage therapy is looking promising, but just drinking the cocktail.
Yes.
Hi, Lauren Cowley, Public Health England. Do you think that when you see silent CRISPR-Cas systems in bacterial strains, that is a function that bacterial strains are looking to increase their genome content with phage infection and prophage regions?
So the CRISPR-Cas is used as a defense against the phages. So sure, it uses it to capture little bits of dangerous phages in that respect. But as far as acquiring like prophages, I'm not sure if there's any data out there that's happening.
But sometimes, do you not see the CRISPR-Cas systems silenced and they're inactive? And so that they were once used as a defense and now not, that there's been a switch so that the bacteria are actively seeking to acquire prophages?
Yeah, I'm not sure about that. That's an interesting question. It's interesting because, for one reason, because many prophages actually bring useful genes to the bacteria.
Yeah.
For example, I didn't mention this, but the cholera toxin genes that cause the profuse diarrhea are encoded on a prophage.
Yeah.
We think of that as a friendly phage.
Yeah.
So that's interesting. I understand now. Bacteria might want to turn off the CRISPR-Cas to allow helpful phages to infect them. Interesting question.
In fact, you can think of them, that's how pathogenicity islands sort of got into the play. They look at, dropping this material in at the tRNA genes, and a lot of it is phage mediated.
Well, the current thinking is that the CRISPR-Cas captures bits of DNA at random, and so once it captures it and then uses it in surveillance, then there's selection on that new strain. And if that captured bit of DNA gives resistance to a lethal lytic phage, then that bacteria will propagate and spread. If it captures a bit of DNA against what would have been a friendly phage, then that bacterial cell and its descendants will be selected against. So natural selection is happening upon these recombinants that capture bits of DNA.
Yes, question.
Hi, Jesse Noah from NC State University. You found that the phage CRISPR system actually could incorporate new sequences into it, right?
Yep.
So I was wondering, considering the limits of the phage genome sizes, do you think there would be a limit on the number that it could capture of new?
Yeah, so some bacteria can capture hundreds and hundreds of these spacers and keep them in their little library, their array. This phage happens to be a big phage, over 100 kilobases. And we don't know how many spacers it could incorporate. But we've seen variants of the phage that have many more spacers than other variants, so there seems to be some plasticity there. But we also see loss of spacers. So new spacers are captured at one end of this array, and you lose them from the other end. It's like a little bit of a conveyor belt. So I think if there was some constraint on the maximum number of spacers, they'll just start losing the oldest ones at the end of the conveyor belt.
Thanks.
All right. Thank you very much for educating us. Now we know a little more crispy information, right? Let's move back. Let's move over to Michele.
Yeah.
Michele, you're working on Legionella pneumophila. Do you remember when you were at Columbia that there was someone working on that?
Howard Schuman.
Yeah. Did that have any influence at all in your
So
working on
I
it?
actually knew Howard Schuman through his wife, who was a postdoctoral fellow at Rockefeller when I was there. And so he actually gave me advice when I was trying to choose a field for my graduate work. And he convinced me that yeast was a great model and I should go work with Marion Carlson and Fred Winston. And as I said, when I was at the Rockefeller, I already had learned about Legionella. So I was excited that Howard continued to work on that or started to work on that at Columbia.
So you're using it now to study cytoplasmic defenses.
Yeah, that's one of our interests. You've probably heard of Legionnaire's disease. You might read about it in the newspaper. It causes these outbreaks where hundreds of people can become ill. But typically only 5% of the people that are exposed become ill. Most of us, we have defenses in our lungs that can capture and clear the organism. So we want to understand how that works, and we were able to use mice as a model system for understanding how white blood cells can detect and then clear contaminants in their cytoplasm. And Legionella is a great tool for this because we're able to manipulate it genetically fairly easily. So we can make bacteria that grow green, or we can delete particular genes. So this story got a real boost when a former MD PhD student of mine, Ari Malosky, made the observation that our wild-type bacterial strain, if you put it into mouse macrophages, it's cleared. But he simply deleted the flagellin gene. So this is the gene that makes the tail that allows these bacteria to motor around in the water. When he just deleted flagellin, now our Legionella strain could grow not only in the macrophages, but also in the lungs of mice. So that led to a series of studies where we and other labs were able to deduce that macrophages are equipped with cytosolic receptors called nod-like receptors that can detect microbial contaminants. So there is a pair of proteins, nod-like proteins in mice called NATE5 and IPAF that can recognize flagellin and then mount a response.
Can I ask you a question?
Yeah.
So the L. pneumophila is taken up into the macrophage by phagocytosis,
That's right.
This is, I'm remembering, Howie Schumann. And
Yep.
then it escapes before that fuses with the lysosome. Is that right?
So that's the other L and A bacterium, Listeria. So Legionella actually stays in a
stays
vacuole,
inside,
but
okay.
it blocks
Resists.
its own delivery.
Tough as hell.
It's tough as hell.
It blocks its own delivery to lysosomes,
Okay.
which is the compartment that degrades. It's the stomach of the cell. It's the compartment that degrades microbes.
So the flagellin is coming from within the phagosome. Is that correct?
So it is coming from the phagosome, but we know that Legionella, like many other microbes, have specialized secretion systems that allow them to inject proteins across bacteria or across host membranes, so they can deliver a payload of virulence factors into the cell. And our genetic data argue that it's during that delivery of payload of virulence factors that some flagellin gets into the cytoplasm, and then it's detected by these nod-like receptor proteins.
So the natural host of Legionella is supposed to be these free-living amoebas. So do they have this cytoplasmic nod-receptor sensing system as well?
So they don't. So Ferric is pointing out that humans never pass Legionella from one to another. We always get it from water. And a large data set from environmental microbiologists and also laboratory microbiologists has determined that Legionella grows very well in amoeba and protozoa. So that's its natural host. And then we become ill if contaminated water is thrown into the air by man-made equipment like the air conditioning system at the hotel in Philadelphia back in 1976 where the namesake outbreak occurred, or more recently at a party at Hugh Hefner's Playboy Mansion where the fog machine got contaminated and there was an outbreak of Legionnaire's disease.
Were you at that party?
I was not at that party.
Ferric course.
I don't remember anything.
I was on the golf course, I'm sure.
Well, that's the other thing is when you look at these golf courses where they're using gray water
Yep.
to water themselves and they're misting in order to do the watering. Do you worry about when you're out on the golf course of getting Legionella?
Most golfers have gone home by the time they turn the sprinklers on.
I always get wet.
So a more common scenario is at the grocery stores, they mist the vegetables to make them look attractive. So if you're a smoker, be careful. Most of us would inhale it and clear it without an incident, but smokers or elderly people could have gotten the organism by that route. Yep.
So Legionella is present in the grocery store.
Maybe not your grocery store, but it has been. It's actually very common in our water supplies.
Where did it come from in the big flower show outbreak? What was the device?
There were fountains that people, you can imagine, these are big flower shows, and sometimes you get bored and just want to just sit down for a while, maybe by the pretty fountain. Yep.
So one of the things that's always intrigued me is Legionella is one of these pneumonias that is only linked with diarrhea. So why does Legionella pneumophila cause diarrhea?
To my knowledge, that's not known, although it is clear that when flagellin is recognized by these nod-like receptors, there's release of pro-inflammatory cytokines, and it could be that if the levels get systemic, and now I'm totally out of my field here, but perhaps pro-inflammatory cytokines
Sure.
could cause diarrhea.
Yeah, that makes sense. It's been suggested for other
Yeah.
viral infections anyway.
Pure speculation.
Yeah.
Well, cytokines are evil, so
Yeah.
Interferon.
when in doubt, just invoke cytokines. You'll get the answer right on the test.
So the flagellins activate the nod-like receptors, and then what happens? How does it get cleared?
So we found that two different things can happen, and it depends on how much flagellin is introduced into the cytoplasm. If it's a lot of flagellin, the cell undergoes this pro-inflammatory cell death that Brad Cookson coined the term pyrotosis to describe. So the cell lyses and it releases these pro-inflammatory cytokines. The microbe can no longer grow. It's exposed to neutrophils that can clear the infection. But we found that at low levels of contamination, we see a different process, but it's still dependent on the same nod-like receptor proteins. And it's a process called autophagy or autophagy, whichever you prefer. Either pronunciation is fine. This is a process, as the name implies, where cells can eat organelles within the cytoplasm. So these can be organelles as large as mitochondria, or they can be phagosomes, perhaps, that a pathogen is in that has been perforated by a specialized secretion system, and then that can be recognized by autophagy. So membranes come from the secretory pathway, and they make a big membrane that engulfs that damaged organelle and then delivers it to lysosomes. So we've been able to show using bacterial genetics and a lot of immunofluorescence microscopy that a little bit of flagellin recognized by nod-like receptors and complexed with caspase-1 increases flux through the autophagy pathway. So this is another example of how autophagy is recruited by the immune system to fight infection.
So some organisms like Listeria and Salmonella can downregulate their flagella.
That's right.
To compensate. Is there any evidence that Legionella tries to shut off this inflammatory response?
Yes, actually, Brian Hammer, my first graduate student, who is now a professor at Georgia Tech and is in our audience, when he was a student with me, he found that Legionella only turns on its flagellin in response to the stringent response. So when the cells become starved, they make their flagella and they swim away from the infected cell. When that flagellated cell is ingested by a macrophage, by the time it doubles, its daughter has shut off flagellin. And Brian showed that using a lovely GFP reporter of the flagellin gene and also antibody. So yes, Legionella does shut off its flagellin, but probably as a economic strategy rather than a evasion of the host. Because to get back to your question, amoeba do not have these nod-like receptors.
Mm-hmm.
So if you disable these mouse receptors, which you can do,
Yep.
does that now make them more susceptible to infection?
That's right. So there's been another lovely body of work done, led by Russell Vance at Berkeley most recently, but also Craig Roy's lab has contributed to this. They've have shown, as has our lab, that if you get macrophages from mice that lack either of the nod-like receptors, now Legionella can grow whether or not it has flagellin. So it's a really pretty genetic story.
So you said only about 5% of human infections lead to pneumonia. So
That's
the
right.
rest, presumably this system is clearing infection, do we know?
So that exact experiment hasn't been done. We do know that human monocytes do have a NAPE gene, and it is responsive to flagellin, so presumably that operates, but the same experiments haven't been done. We also know from early work from Marcus Horowitz that once our macrophages are activated by gamma interferon, they're now restrictive to replication. And interestingly, gamma interferon is also known to induce autophagy. So it's possible that a couple of these mechanisms contribute to restricting replication in the lung.
This morning at the meeting, I heard a lovely talk from Jean-Laurent Casanova at the Rockefeller, where he came to view infection as a sign of a problem with the usual commensal balance between a bacterium and the host. So he said that all children are colonized with pneumococcus, but only a small fraction get invasive disease. So when he, as a pediatrician, sees that disease, he wants to know what's wrong with that kid.
Right.
And that has led him to identify all kinds of new mutations in essential pathways that defend us against different kinds of pathogens. And he was showing how they had, over a period of 20 years, found this specific mutation that was the cause of the lack of a spleen in development in a child who would then manifest as an invasive pneumococcal infection. So with Legionella, since we're being exposed all the time, but only a small fraction, some of those people I know are elderly or they have immunosuppression from... So I'm just wondering if there are also genetic predispositions that have ever been described in humans to make them more susceptible to Legionella.
Yeah, so actually that flower show outbreak in the Netherlands was a large enough outbreak that it was a good opportunity to ask that question. And it was found that there was a slight risk, increased risk, by people who had mutations in a toll-like receptor 5, that sits on the macrophage surface and also recognizes flagellin. So that may be...
was it a gain of function or a loss of function mutation?
I believe it was a loss of function mutation.
So that might have changed the balance of signaling in response to flagellin.
That's right.
Interesting.
So other pathogens, as you know, tie in with the autophagy pathway. In fact, the virus I work on, the cells initially turn on autophagy, and then the virus actually utilizes it to get out of the cell, and it blocks the fusion of the autophagosome with the lysosome, so it doesn't get degraded.
So we're actually very interested in that work because when we look at macrophages and just treat them to starvation to induce autophagy, we actually see quite a lot of evidence that autophagosomes are also fusing with the plasma membrane. So we're interested in whether this is not a mechanism that some viruses exploit to spread, but perhaps the host uses to capture cytosolic antigen and deliver to neighboring antigen-presenting cells. So that's an area we'd like to explore.
Does it make any sense for Legionella spread?
No, it doesn't.
Because it gets out other ways. You're looking at me like I'm crazy. I'm just asking dumb questions, that's all. So tell us one next step in this story for you.
Well, as I mentioned, we've discovered by looking in the microscope that we do see autophagosomes going to the surface of macrophages, and we'd like to understand how that might be contributing to immunity.
In one of your papers, you mentioned a connection of this with Crohn's disease.
Right.
What is that?
So Crohn's disease, as you may know, is an inflammatory bowel disease, and it affects enough people that large genetic studies have been done. And interestingly, they've identified some similar components that we have found to be important in Legionnaire's disease. So NOD-like receptor proteins and also autophagy proteins are important for keeping your gut healthy, because mutations in either of those classes can increase a person's risk of Crohn's disease. So the point I would make then is that by using bacterial genetics in a very simple mouse model, we can really study mechanisms of cellular defense and relate that back to the more complex story in the gut of IBD patients. And hopefully our work will inform studies of Crohn's disease treatments.
Thank you, Michele.
Thank you.
Let's move to the eye. I bet you've been waiting. I appreciate your patience. I was intrigued by your story because we don't really think about the eye much on TWiM, right?
No.
Not at all. And you use Pseudomonas as a model to study actually why the eye is so resistant to infection, right?
Yeah, actually, you raise a very interesting topic, and that is why we ignore the eye. It is a part of the body, and I find this happens a lot. I mean, it happens in the field of microbiology. And I remember when I was at Harvard and I was teaching in the program, in the infectious disease program, and we would be talking about microbes that actually the most common site they infect is the eye, and yet that aspect wasn't in the curriculum. And I remember talking to people about that, and they'd say, well, it's a specialty area. But it's an interesting concept, and we see this in the journals too. There are eye journals, and a lot of the eye researchers publish all their work in the eye journals. So that's interesting. Actually, in the end, I think it's turned out to be an advantage for eye research, because what's happened is it's become a very multidisciplinary field. So you have people who do microbiology, immunology, cell biology, optics, different types of physics, and they all get together and have conferences like this. But instead of being focused on one discipline, there's all these different disciplines. So we're very accustomed to talking to each other and have knowledge about all these other areas. So I think that's been an advantage for eye research that now has propelled research in eye disease in some areas ahead of diseases of other
You really
sites.
understand your host, because the eye being immunoprivileged, it really is a host unto itself. In order to participate in it, you really need to understand the host. Whereas the rest of us, the host is this gigantic, complex thing called a person.
That's right. And I think it's also helped me in terms of imaging. So, you know, we use a lot of imaging in our research, and in my building, there are researchers that do optics research who have been able to help us to set up our systems as an example. But, you know, I think that's been useful. So, yeah, I use the eye as a model for studying the question of why we don't normally get infections. So this is something I think that we haven't really done much on. And, you know, I've pondered the question of why that is the case over the years, and I think that actually studying why infections don't happen is more difficult than studying why infections do happen, and that's because we have all these infection models, you know, where we make an infection and then you actually have something to study. But studying why infections don't happen, it's much more difficult to develop those models. So over the last few years, we've actually been spending time developing such models where, you know, we look at, well, what happens when you put a lot of bacteria into an eye? You don't get an infection. So why is that? You know, what are the defence mechanisms that operate to protect us? But, you know, really it's the same thing anywhere else. So we have, if you take opportunistic pathogens like Pseudomonas and introduce them to a healthy host, they don't get an infection. And so it's not really just the eye that is resistant.
So when you talk about putting bacteria in the eye, whose eye are you putting it in?
Not people.
Right.
So we do use human subjects in our research as well. And actually this is, you know, in my talk yesterday, I spoke about some of the advantages of doing eye research for studying infectious disease. And another one is that you can actually collect cells from a person very easily. So we have a piece of equipment, it's called an irrigation chamber, and it's like a miniature drinking fountain. And you can use it to rinse epithelial cells off the surface of the cornea, and they're still viable, and you can actually study those in experiments. The tear fluid is actually a model for mucosal fluid. So we collect tear fluid from people. So we can actually design studies with human subjects and collect material from those people and then do in vitro experiments using the materials collected from people.
How do you collect tear fluid? In dentistry, they give you some wax to chew on to get saliva. Do you make people cut onions?
No, I make my graduate students cry.
Oh! Graduates,
I torture my graduate students and postdocs.
She makes her graduate students cry.
No, actually, no, we're not allowed to do that.
That's true. It's the kinder, gentler world. We can't beat our graduate students any longer.
Yeah. Well, there's several different ways you can, a human subject, have them cry. Some people like to think about something sad. Onions work.
Onions work.
Also, the procedure that you use to collect the tears, it doesn't hurt, but it just irritates a bit and it can make the eye tear. And so some people are good tearers, you know, good criers, and you get a lot of tears, and other people, it's more difficult.
Are there any host defenses that are unique to the eye?
You know, I'm starting to think maybe not. So one of the things that I think is different actually about the ocular surface, and so the ocular surface actually doesn't have commensals. So if you culture the ocular surface, you will get, you know, a few colonies of this and that, but that's because it's exposed to the environment, and so things are constantly falling in. Now, you know, our experiments with Pseudomonas have really shown us that the ocular surface zaps bugs. So if you put bacteria in there, they disappear very quickly. So even Pseudomonas, which is, you know, one of the most difficult things to kill, just gets zapped very fast. But I actually think that what may be going on is that the reason you don't have bugs there is that the epithelial surface is probably, and it's probably the same, all of our epithelial surfaces is antimicrobial. And so the reason that you don't have bugs on the ocular surface is because the tear fluid is so thin that it's not possible for microbes to get far enough away from the epithelial surface.
And what is it about the surface? Does it make peptides, or are there?
Well, if you take corneal epithelial cells and you grow them in culture, they behave just the same as any other epithelial cell. They do everything the same way. They have all the same features. We haven't really seen very much that's unique about those cells. So, and then the tear fluid is a mucosal fluid. And I don't know whether there's anything particularly unique about that either compared to other mucosal fluids. But I think the thinness may be critical because, it's been shown in the gut that there's a clear zone between the epithelial cell surface and where the commensals hang out. And it's about 50 microns. So the tear fluid is only about 7 microns thin. So if you translate the exact same situation to the ocular surface, there's going to be no bacteria there.
Is it nutrient poor, tear fluid?
No, it's actually nutrient rich. And actually, if we take tear fluid out, we can, about 50% of the Pseudomonas isolates, clinical isolates that we've tested, will actually grow in tear fluid.
It seems really paradoxical.
Well, was it blinking? Does blinking
blinking obviously physically will contribute as well. But I actually think it's that. And actually, if we take corneal epithelial cells and grow them in culture, if you inoculate them with Pseudomonas or any bacteria, and I think it's the same for any epithelial cell, you look down the microscope, you never see bacteria actually attaching to the cell surface. What they'll do is they'll swim a certain distance away.
The GI tract is kind of analogous. I don't know if it's by the same mechanism, but you have these cryptins that are produced in the intestinal crypts that create a bacterial free zone right at the site where the new cells are being replicated and produced. So you'll see a huge number of bacteria, of course, with the stool, but there's a discrete distance in a healthy intestine between that and the crypts. But I thought I understood that because there are all these antimicrobial peptides being produced.
So the cell does make antimicrobial peptides, the same, but I think it's the same as other epithelial cells. So I think all of our stratified squamous epithelia do this. They release antimicrobial factors, and that's why we have the clear zone. And then in the eye, we happen to have a very thin layer, so it's all going to be clear. And I think this actually gets at the reason why. So we've been studying why contact lens wearers are predisposed to infection, and I actually think that's one of the reasons why contact lens wear predisposes to infection is that you're introducing an object that the microbes can actually grab onto, and they grab onto the back surface of the lens. And in fact, we developed a rat model, so we got contact lenses for rats.
Oh, God.
And what we did was we actually inoculated them with Pseudomonas and then put them on the rat, and every single rat got infected, every rat. But the really interesting thing was that it took an average of about 8 days for the rats to get infected. So during the 8 days, what was happening was a thick biofilm was forming on the back of the contact lens. And then, we think that the bacteria actually are adapting to the host environment in that biofilm, and that then enables them to infect. But another interesting thing that we saw was that when we took the lenses out, the back surface was just covered in, really dense biofilm, but the front surface was completely clean.
So these are extended wear rat?
Yep.
Did you find the rats behave differently when they could see what you actually looked like?
Don't know. Well, these had no optical power, so. Yeah.
A lot of contact lenses cause some ischemia also. That ophthalmologists talk about that. I wonder if.
Well, okay, so I've been working on this for a long time, this problem of why contact lenses cause infection. And so I've been working on it so long that I predate the introduction of silicon hydrogel contact lenses, which everyone's now wearing. So these are the ones that allow through physiological oxygen levels. Okay. And when the companies were working on this and they were all claiming it was going to solve the infection problem, I didn't think it was going to. And actually, I still have this New York Times article from when the FDA was about to approve these. They actually interviewed me, and I said that I didn't think it was going to work.
Have you tried the new lenses in your model to see?
No. However, the experiment's been done on people because the silicon hydrogel lenses have been around long enough that the comparisons, the epidemiological studies have shown there's absolutely no difference in the infection rate, and it hasn't changed the spectrum of microbes that cause the infections either. So it's still Pseudomonas number one, causing about 60% of the infections, and then a splattering of other microbes causing the rest. And in fact, the only one difference that's really been seen is that inflammatory complications have actually increased.
With the new.
So it's another complication of contact lens, whereas to get a sterile inflammation, which may actually relate to microbial ligands, even though there's no live bugs. And they're seen more commonly in people who wear silicone hydrogel lenses, which allow through physiological oxygen levels.
Do you wear contacts?
No.
You would never wear them?
Well, I'm not at the point where I've had to worry about making that decision yet,
But if you did?
if I did, I wouldn't wear them on an extended wear basis.
Okay.
Based on the fact that we think that this, the biofilm formation combined with extended exposure and adaptation, and I think the adaptation part of this probably explains why Pseudomonas is the number one pathogen, because it's so good at adapting to its environment. And so I think if you take the lens out every day, clean it,
rinse it,
and better still, wear a new lens every day so you can get daily disposables. Mm-hmm. So you take a fresh, clean lens out every day. Now, sometimes you still get infections with those, and it may be that the whole process is sped up on occasion in some people under certain circumstances, but the incidence of infection is much reduced with daily wear.
Now, acanthamoeba is associated with contact lens wear and doesn't form biofilm, so what's the connection there?
Well, I think that's very different.
Because we must be bathing our eyes with acanthamoeba all the time when we take a shower and so on.
Yeah, I think it's a different story. Look, I think there is another part to the pathogenesis of contact lens infections, and there certainly is a suppression of innate defence as well. Mm-hmm. And what is interesting is that all of the big bugs that cause contact lens infection are all opportunistic pathogens. Yeah. So they tend to be the bugs that are out there in the environment that we're constantly exposed to.
Do you think is there trauma from the contact lens to the epithelium that allows them to
very little trauma, and in fact in our research we've actually shown that you can induce quite significant trauma to the cornea and still you won't get an infection. So you've got to go to quite desperate measures. You've actually got to completely obliterate the epithelial layer all the way into the underlying stroma. If you have anything that's more subtle than that, the eye will still clear all of the bacteria out.
Is there any way to prevent biofilm formation? Is that something that...
Well, I guess, there's other people working on that.
So you would have to put something in the lens itself, right?
Yes, I think that would probably be a very good way of eradicating contact lens-related infections, is to have a way of preventing the biofilm formation on the contact lens.
What are the numbers? How many infections in long-term use?
In extended wear, it's about 1 in 500 per year.
Whoa.
A lot.
So let me ask you this question.
And these are corneal infections, which are sight-threatening.
Mm-hmm.
So one of the intriguing things is all this new laser surgery that is being argued, and as we all are getting older with each passing day, some of the new laser surgery will get rid of the need for reading glasses. Has anyone begun to study how the microbes will interact as you begin to ablate the cornea and tinker?
Well, these procedures have been done for quite a long time. I think it's a terrible thing to do to your cornea, because you're basically taking a healthy cornea
and you're
destroying it. I have a lot of respect for this tissue since I work on it. So it's not something I would have done. My big concern is, so when you have the procedure done, you can be susceptible initially before the healing happens. But the other thing that can happen is, they usually make a flap. They cut the cornea and burn under it, and it smells like hair burning if you stand next to someone having it done. You can see the smoke coming off. And then they put the flap down. Now that flap, it never actually completely attaches. So you can actually,
oh God.
You knock
so
your eye
you go
in
on
the
a
right
roller
direction.
Coaster, you'll go blind.
You can actually come off. But I'm also very worried about, so there's a layer of cells on the back of the cornea that's a single monolayer, and they're permanent cells that don't replace themselves, and they're critical for function of the cornea. They keep the hydration level correct, which is important for the optical properties. And, I worry about the laser being so close, the high energy laser, so close to those endothelial cells, and what effect that's going to have on people in the future. So, these procedures have been around for a while, so we know that, in the short term, there's still a lot of complications, but I worry more about someone who has this done in their 20s and they live to 80. What is their cornea going to look like at that point?
So when you do get a Pseudomonas infection, what are the complications? Blindness, I presume, is one?
Well, if Pseudomonas gets into your cornea and the disease begins, it's really fast. Like it melts the cornea.
Okay.
So
you do not want to see that seminar at lunch. The ophthalmic surgeons and people just like you show these pictures, and it's frightening.
Yeah, and sometimes if the infection happens in the periphery of the cornea, it tends not to blow out of control as badly. And it depends how quickly people seek treatment, but it's pretty quick. Once it gets going, it's very painful. And then, unfortunately, if it gets past a certain point, it disrupts the collagen structure of the stroma of the cornea, which is critical for the clarity of the cornea so that you can see. And once that's disrupted, it doesn't heal in a regular way, and so you end up with scars on your cornea.
Okay.
And if they're in the centre of the cornea, it can be devastating for vision.
Right. So if you don't wear a contact lens and you get a Pseudomonas eye infection, why is that? A scratch or something?
It hardly ever happens otherwise. So the only other circumstances that people get Pseudomonas infections in the cornea is if they have a deep penetrating injury to the cornea that goes all the way into the stroma, which is pretty unusual. Now, what's interesting is that in India, there's a fairly high rate of Pseudomonas corneal infections in people who don't wear lenses. And, as to why that is, no one really knows. But it's a very, I've never been to India, but people who've been there say that it's, the environment's there's a lot of dust and pollution and wind and it's just bugs everywhere. And so, I think that's a whole different, it may be a different pathogenic mechanism there.
Right.
I'm going there in November. I'll
With
get back to you on
sandstorms in the Middle East, with the debris coming into your face if you're not protecting your eyes.
Yeah, I haven't heard any reports of...
I've heard about the Indians, but I have not heard about, and I think it's one of those great mysteries.
But, I think it's an interesting thing to ponder because it might actually, there may be some clues as to why Pseudomonas causes infections.
Yeah, this is maybe out of your area, but there are certain avian influenza viruses. When there are outbreaks on chicken farms, often the workers, their conjunctiva are infected with the virus because it has receptors for avian influenza virus strains, alpha-2,3 sialic acid receptors. And then from there, the virus can drain into the respiratory tract.
The conjunctiva and the cornea are quite different.
Yeah.
So the cornea is an immune privileged site. It's avascular, and so it's kind of separated from systemic immunity. And the conjunctiva is a whole different story. So there's conjunctival associated lymphoid tissue, and so it has a close connection with the immune system. And so you tend to get infections with different microbes in the cornea and in the conjunctiva. And actually a lot of the conjunctival infections are inflammatory in nature.
Right.
So it's not actually, well, it is an infection and it's caused by a bug, but the disease itself is inflammatory.
Right.
Do you think that the immune privilege of the cornea is really by design because any immune response would be so damaging to function? Kind of the way in the lung.
That's exactly the case, yeah.
Reason.
So all of the components of the eye that are critical for vision are immune privileged. And for the cornea, part of it too is that you don't want to have blood vessels there. So there's no blood vessels in the cornea because otherwise it would obscure vision. Yeah. And also there's very few immune cells for the same reason.
Well, that was great, and I thought it would be cool to have a former optometrist. Now, are you a microbiologist now? Is that what
I have a PhD in microbiology, so I guess I am, yeah.
Remember, it is the advanced degree.
Yeah.
Yes, of course. And I learned a lot about the eye, so thank you very much for joining us today. And that will do it for this episode of TWiM, which you will be able to find at Microbe World, there's a B in it, .org/TWiM, and also on iTunes. And if you like what we do, one thing you can do to help us is to go to iTunes, where our episodes are listed, and just leave a comment or a rating. And that helps to keep us very visible in the iTunes directory. And we usually answer email questions on TWiM. We don't have time for it today, but you can send those to twim@twiv.tv. I want to thank all of our guests for joining us today. Michele Swanson, the University of Michigan, thank you.
My pleasure.
Joining us. Ferric Fang at the University of Washington.
I enjoyed it very much. Thank you.
Elio Schaechter from Small Things Considered. Thank you, Elio.
Of course.
Did you enjoy doing this in vivo?
Yeah, it's not bad.
Very good. Michael Schmidt, Medical University of South Carolina.
Also in vivo.
It's so in vivo. It's not often that we are in vivo with each other. Thank you.
No, thank you.
Suzanne Fleiszig, because I say Fleiszig. Suzanne Fleiszig, University of California, Berkeley. Thank you for joining us. And Andy Camilli from Tufts University.
Thank you.
Thank you
Enjoyed
very
it.
much.
Did you bring Elio his letter opener back?
No, I still have it and I still use it.
Where's my...
I have a similar story. I have a former student who has my stapler, which I took as a graduate student from Mount Sinai. So this is part of science. I want to thank the American Society for Microbiology for supporting TWiM and letting us do this at this meeting. We really appreciate it. In particular, I want to thank our producer, Chris Kandayan, over there running the TriCaster, which is a very cool piece of equipment. You should check it out on your way out, but don't step on it. And also, Andrea and Ray behind the cameras, thank you very much. The music that you usually hear on TWiM is composed and performed by Ronald Jenkins. And you can find his work at ronaldjenkins.com. I want to thank everyone for listening. We'll see you next time. Did I say who I was?
No.
I didn't. Forgot to say that. I'm Vincent Racaniello, and you can find me at virology.ws. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.